Okay, welcome to our first Hangout live on air with the Australian Society for Parasitology. And this is an Inspiring Australia public outreach initiative. And we've got some parasitologists here from across the world, and they're going to tell us a little bit about their work and their research and what makes it fascinating, and also how they became involved in social media. So and sort of different ways that we can use social media to talk about the research that we do and promote the research that we do across to the wider community. So I guess I'll pass it over to each of you to introduce yourself. Tommy, do you want to start the ball rolling, and then we'll move over to Kelly and David?
Sure. My name is Tommy Leung. I'm a lecturer in parasitology and evolutionary biology at University of New England in Australia. And I work on a variety of different parasites, but just by kind of coincidence, most of the parasites I've worked with over the years are parasitic flatworms, such as flukes and tapeworms, mostly larval stages in invertebrate hosts, and mostly aquatic invertebrate hosts, so crustaceans and mollusks. So that's the parasitologist that I am.
My name is Kelly Weinersmith, and I'm a PhD student at the University of California, Davis, and I study Euhaplorchus californiensis. So this is a trematode parasite that infects the brains of California killifish and causes them to do conspicuous behaviors that make them very obvious to predatory birds, which are the next host in the life cycle for the killifish, or sorry, for the parasite. And these conspicuous behaviors appear to get the killifish eaten by the predatory bird and completes the life cycle for the parasite. And I look at various ways that the parasites manipulate the physiology and behavior of the killifish host.
Hi, my name is David Hughes, and I'm an assistant professor of entomology and biology at Pennsylvania State University in America. I'm interested in parasites that live inside insect societies, so inside wasps and ants. I've worked with a few different systems. I initially began my PhD work on a rather curious group of parasites called Strepsiptera, which are an order of insects and have rather bizarre biology, and I hope we can get into some of that later on. And then went on to other systems. I worked a little bit on hairworms, which control not social insects, but solitary insects, crickets, causing them to jump into bodies of water where these large worms can emerge. And for the last 6 to 8 years, I focused almost exclusively on fungi that control the behavior of ants, the rather well-known system now of zombie ants. In this case, again, an example of parasite manipulated behavior, but rather unusual because it's caused by an organism which has neither a central nervous system nor any muscles of its own, and it co-opts those of its host to get from A to B. And so that's my general interest in parasites. Those parasites control animal behavior.
Excellent. So, Tommy, can we go back to you and just, do you want to tell us how you got into parasitology in the first place, and then we'll move along across everyone as well. What sort of inspired you right at the start? Were you, you know, like, and what was maybe the first parasite that took your fancy, and have you continued on with that relationship?
Well, it wasn't a parasite exactly that took my fancy, as in something infected me. No, I was infected by an idea, which was when I was in second year undergraduate studies, I read a book by Carl Zimmer called Parasite Rex. And rarely can you say that like one book kind of changed your life, but in this case I can say definitely that I read Parasite Rex and I thought parasites are very, very interesting, and most people really overlook them. So that's when I decided to I basically gave myself a crash course in parasitology, even though for my undergraduate degree I never learned anything about parasites except that someone might have mentioned it in passing in an ecology unit. I went out and I searched the literature and read everything I possibly can on parasites, and that particular interest and enthusiasm has kind of continued to this day.
Kelly.
So I have a similar story, actually. I read Parasite Rex, and I thought it was really cool, but I didn't immediately dive into parasites. That just sort of made me more excited about parasites. And then one day my advisor, Andy C., sent me a bunch of papers about how humans that have toxoplasmosis have different behaviors than uninfected humans, and how this parasite seems to subtly be changing human behaviors. And that blew my mind. I thought that was awesome. And then I contacted the Curious Lab, or the Ecological Parasitology Lab at the University of California, Santa Barbara, to tell them that I was interested in looking at how personality traits changed when hosts were infected with potentially manipulating parasites. And they agreed to let me be a visiting scholar in their lab for a couple years, and I learned about my study system, and now I am just totally obsessed with parasites. Yeah.
For my part, I was introduced to parasites through the very wonderful Bill Hamilton, who was my supervisor for a few short months before his rather untimely death. And Bill had discovered, Bill Hamilton, if you don't know, for those out there, is the person who discovered kin selection and resolved Darwin's one special difficulty. So in writing The Origin of Species, Charles Darwin was perplexed as to why ant societies had sterile individuals, and he came up with a solution, but it was only a partial solution. And it was Bill Hamilton who laid down the framework, a mathematical framework with kin selection. And so upon his death, Richard Dawkins, writing an obituary for him in the Times, said he was probably the most important biologist since Charles Darwin, certainly a very important naturalist. So Bill was my PhD supervisor, and I was very fortunate for that experience. And the reason he was a supervisor of mine was because just before I began my PhD, he discovered that large numbers of social wasps, paper wasps in Italy, were deserting their colony at the very height of the season when they should all be working industriously. And the reason for this desertion was because of an endoparasite called Strepsiptera. And so you can't see the parasite, it's rather cryptic, and indeed this is the very best studied system of social insects, or wasps at least, anywhere in the world. And for 50 years, researchers had worked very industriously on the system, and nobody had discovered that large portions of the population were moving away because they were being controlled by a parasite. And the reason they moved away was because the parasite needed to reproduce, and it couldn't do that on the nest itself. And so Bill's view of this was looking at the system through the eyes of the parasites, and that's how I came to parasitology, and how I've come to all the systems I've worked on, and I'm rather grateful for that. So I say my beginning was with one of the very best, Bill Hamilton.
That's amazing. You seem to all have an interest in that parasites affecting behaviour of their hosts. And are you still, so David, are you still pursuing that as, because Kelly, you're still pursuing that as one of your main research areas. And David, are you also?
Yeah, so this is truly my main research area. I'm interested in fungi to control ants to leave their colony. I've done a lot of work in tropical forests on tanking this system. And then over the last couple of years in the lab here at Penn State, we've moved towards a focus on the proximate mechanisms as well as the ecological significance. So we have these different strands going on. The proximate mechanisms is work that's ongoing in the lab, and Shrissa de Becker is a really excellent postdoc in the lab working on this system. In the ecological scenario, we do a lot of work in the Amazon rainforest. And I think the work that we're doing is reflective of what's ongoing in the community. People are moving beyond the coolness of the systems to try to ask what's the significance there. So yes, I'm very much focused on parasites that control behavior, and trying to explore from both the small steps with proximate understanding, as well as a larger ecological significance across continents.
Kelly, can we move on to what you were, your current study that you're doing at the moment, and could you maybe outline as well in what your research is doing that, you know, or the current research that you're doing at the moment, and that's also to do with parasites affecting the behaviour of different animals, of their hosts.
Yeah, so as I mentioned, I study that brain-infecting trematode parasite, Eueplurchus californiensis. So it infects California killifish, and then it moves up to their brain. And we've noted changes in brain chemistry following infection, and those chemicals in the brain that are changed, specifically their serotonin and dopamine, the concentrations and the turnover of these neurotransmitters are changed following infection, and that's associated with a lot of different behaviors in fish. And these behaviors include activity and boldness and exploration. And so I'm interested in knowing if these killifish have essentially completely different personalities when they're infected than they would if they were uninfected. And we've already noted that these killifish, when they're infected, seem to do more of what we call conspicuous behaviors. So these are things like they'll turn upside down and they'll flash their silvery bellies up towards the sun, or they'll shoot forward really quickly, or they'll contort their bodies. And actually, infected schools of fish look more like they're dancing through the water than swimming. And they're very obvious to human observers, and so you can tell if there's a bunch of infected fish because there'll be these bright flashes of light as the school goes by in an otherwise dull estuary. Well, the estuaries are interesting, but the water, there's not much else going on. Anyway, so I am measuring the behavior of these killifish prior to infection. So I got a bunch of uninfected fish, and then I infected half of them, and I'm measuring their behaviors again to look to see do the conspicuous behaviors increase following infection, because previous work that we've done has looked at naturally infected fish, and we can't be sure that these fish that do a lot of conspicuous behaviors aren't the ones that maybe they happen to get infected because these conspicuous behaviors put them in contact with the parasites.
Yep.
It seems unlikely, but you need this manipulative study to be sure that it's the parasite that's causing it. And so we, by the end of the study, hopefully I'll know if these fish are more active and more bold and more exploratory and more likely to do these conspicuous behaviors. And so that's what I'm working on at the moment.
So your premise as well is that the fish are, or the parasite is just trying to get to the next stage in the life cycle. Is that the...
Yes, yeah. So there was a study that put infected and uninfected fish in the same outdoor enclosures, and this study was done by Kevin Lafferty and Kimo Morris. And they found that the infected fish were 10 to 30 times more likely to be eaten by predatory birds than were the uninfected fish. And predatory birds are the definitive host in the life cycle for the parasite. So we think this is the parasite's way of getting the attention of predatory birds so that it can complete its life cycle.
David, do you have that same experience in the study that you're doing, that the parasite itself is just trying to make its way through the life cycle, and that's why it's causing the host behavior?
Yeah, so I should have specified exactly what my system is. We have ants in a society, in a nest, and they go out to forage in the rainforest. And as they're traveling in the undergrowth, they pass through areas of high densities of fungal spores. These spores attach to the ant's cuticle or their skin, and then they literally blow a hole through the ant's body and then replicate inside its body over a period of 2 weeks. At this stage, the ant is back inside the nest, surrounded by many thousands of other ants inside the colony. But the fungus, which is the parasite in this system, is unable to transmit inside the colony. And the reason for this is the social immunity, which is a distinguishing characteristic of ant nests. They're incredibly clean and sterile places, and they need to be because they exist with as high density aggregations of many thousands or sometimes millions of individuals, all highly related. So in order for insect societies to exist, they need to necessarily sort out the problem of disease passing from individual to individual. But the parasite is specialized and needs to transmit. So what it does is it causes the ants to die outside the colony. And it typically causes them to die on the underside of a leaf, thereby providing a platform. Because what's going to happen once the ant dies is that the fungus is going to transition from growing inside its body to growing outside by forming a very large stalk that stakes out of the back of the ant's neck down, and then from that it produces a football-shaped structure from which spores are produced and shot down, and I like to say that can also be an Australian football. And then these spores launch up the ground, and then the ants become infected. So the manipulation is going out of the colony to a leaf, biting onto a particular part of the leaf, the main vein, and all of this happens with a beautiful synchrony around about solar noon. So this is one of the most complex examples of parasites controlling behavior that we know about, because this is not within the normal repertoire of these ants. They don't normally bite into leaf. They have no business doing so. And what's also rather startling in this system is that the forest is large and it's full of leaves. So the fungus needs to necessarily direct its ant host to a particular set of them. Too far away from the colony and you won't be able to transmit. Too close and you are in danger of being cleaned up by the large numbers of individuals which have this rather specialized hygienic behavior.
It sounds like a horror movie. all of the
Yes.
studies that you're doing. Tommy, do you want to tell us your research in particular, and is it also sort of looking at behaviour of parasites?
Well, I started out. Oh, sorry. Well, I started out looking at behavior, but I've kind of moved on now to looking at interactions between different parasites when they share the same host. Because during the course of both my PhD study and my postdoctoral study, and now, I often find invertebrate animals, even though they're quite small, you can often find multiple species sharing the same host. And most of the question that I've become interested in is how different species, often with conflicting goals for what they want to do with the host, how they either live with each other or don't live with each other. So one of the studies that, for example, that I did during my postdoc is to look at snails that live in the intertidal ecosystem, and they often become infected by parasitic flukes. Now, parasitic flukes, they go through a complex life cycle, and at one stage of their life cycle, they infect a snail, and inside that snail they undergo asexual proliferation. So they have one tiny little larval stage that then proliferate, and practically it takes over the inside of the snail's body. It takes up between a quarter to up to even in some case half of the biomass inside the shell of the snail. And they use this they use the snail as a kind of a parasite factory. So it then starts pumping out clones of these larval stages, which then go on to infect the next host of the life cycle. In the case of the species that Kelly works on, fish, and in other species it infects a crustacean or maybe even a mollusk. Now there's limited resources and limited space within the body of the snail, and sometimes the snail can become infected by multiple species, 2 or even in some case 3 species of different trematodes. And one of the projects that I looked at for my postdoc is to look at a particular species where they have the asexual stage that seems to display some degree of morphological differentiation. So you have the stage that produces the larval stage that swims out of the snail, and then you have a separate stage which looks very, very different. They're very tiny, but they have a big mouth and a big gut. And the inspiration behind that was actually when I met someone from the Curious lab, Ryan Hetchinger. He noted that there are these little things inside the snail called what he calls soldier casts. So these are trematode flukes. They're asexual stage, but unlike the stage which are large and sac-like and produce the larval stage that swims out of snails, they're tiny and they're very active. They move around inside the snail, attacking any co-infecting species. And when he told me about these guys, I started noticing, hey, there is a species that is just like it that exists in the New Zealand system where I was working at the time. So in addition to that, I also look at other systems where there are co-infecting parasites sharing the same host, and how they either benefit from that kind of relationship, or in some cases it's either detrimental one way or another.
Yeah, it must have a burden, or the burden on the host must be more if you've got the more parasites you've got in there. Surely, you know, are they all competing for the same bit of the host, or are they do they sort of live in different parts of the host?
It depends on what species. So I guess with these parasitic flukes, they all share the same part of the snail, which is usually the gonad and the hepatopancreas, which is a kind of an organ which is a mix between a stomach and a liver. However, in other species of invertebrates I've worked with, so for example, during my PhD, I worked on a bivalve called the New Zealand cockle, which is actually a clam. And in that case, you have a parasitic fluke larva that lives. Some of them live in the mantle cavity of the clam. Some of them live inside the foot of the clam. The species that live inside the foot of the clam actually does some degree of behavioral changes to the clam, not through any sophisticated kind of behavioral manipulation as seen with the flukes that Kelly work on or the fungus that David work on, but just by being there. So the presence of the parasite presents kind of a physical or a mechanical obstruction. And when you have hundreds and hundreds of these parasites building up in the foot of the cockle, eventually the cockle loses the original function of the foot. And the original function of the foot, which is to act as a kind of a muscular digging organ, what happens is that often these clams, because they are only living underneath the sand, a few millimeters underneath the sand, they often get washed up on the surface of the beach. And so when they get washed up, they have to stick their foot out to dig themselves back in. But if that foot becomes filled up with parasite tissue, then it can no longer do that. And the next stage of the life cycle for this particular parasite are for seabirds, such as oyster catchers or seagulls, to come along and eat the cockle. And so the parasite benefits from crippling the host in this way, but there are also other parasites that undergoes the same life cycle, but they don't go in the foot, they go in the other part of the cockle. And they actually benefit from having the presence of this species that goes to the foot, because it kind of almost, they are almost hitchhikers.
They're hitchhiking onto the next host.
Yeah.
Very interesting creatures, aren't they? So we'll just move on to the last part, which is about the social media activity, I guess, as well. And so you're all very active on social media, bloggers, and also, you're just engaging in Hangouts and things like this. Can you just give us a little bit of an insight as to, I guess, how did you start it? It's pretty complicated for somebody who hasn't done it before, but what stimulates you into, keeping on doing it? Does it get easier as you do it more often? And does it help with your research? Is it just good fun? Do you feel like you're sort of also doing sort of a public message about parasites? And, sort of just give us a little bit of an insight into your own experiences, I guess, in terms of being involved with social media and parasites. David, do you want to start?
Sure. I think Kelly's so much better at this than I am. I'm very much following her footsteps here. So I don't blog, but I do take part in Twitter, and I enjoy that immensely. And I've also made some YouTube videos which have been quite popular. I have done an awful lot of social media through another endeavor of mine, which is called Plant Village, which is connecting together people in a large community to share knowledge on growing food plants. So, of course, the web is absolutely revolutionizing the way we can contact other people, and I rather liken this to how we might interact at a conference. I'd rather been impressed by how I will meet people at a conference every couple of years. I might not see them for a few years, but then once we bump into each other again, it's like we've never left one another, and we can drop into easy conversations. And I think Twitter and the ability to connect people through their emails on blogs it has that sort of conference feel about it. It's very, very relaxed and very, very informal. And so Tommy, for example, has written something on his blog about my parasites, and it was very nice to engage with him on Twitter, and then it's led to something like this evening here, which hopefully somebody out there watching it will learn a little bit more. And it's just another way for us to engage with the public, with people who are interested. I know for me, growing up as a young kid, I got biology through David Attenborough every Saturday evening, and that was really beautiful. But maybe this is not the way kids are finding biology these days. Maybe they're finding them in smaller snippets. So through the web, we can produce those snippets, whether they're YouTube videos or other things that I've done. I've been a part of a Sony PlayStation game called The Last of Us, as well as the Paramount movie World War Z. And in both of those cases, I've made small documentaries. And I think these small snippets have a life of their own on the web. And I think this is what social media can bring to us. It's very much a new ground. I don't think anybody really understands it, but it's very much fun to engage in. Absolutely, I enjoy. But let's give the word over to Kelly, who's so much better at this than I am.
Well, so I got involved in social media at first, partly selfishly, just because I really love talking about science, and the idea that I could have an audience for that just seemed exciting. But then I was asked to be a guest on a podcast called Science Sort Of, and I really enjoyed talking with that group of people. And the idea behind that podcast is everyone gets together over a beer and talks about a couple different science articles or interviews someone. And I really enjoyed that, because if I were to be deciding what I would do on an evening off, that's what I would want to do. And so to be able to just have that sort of fun conversation and then put it out there and then get feedback from people and hear that made them excited about science and they learned something new, that was just a really rewarding experience. And so I'm a regular on that podcast now, but I also started my own podcast with my husband, And that mostly started because he and I talk about science for at least an hour every week, and we figured, well, we might as well record it and share it with people, and it also is a great excuse to connect with other people. So we both read this book called The Other Brain, which is all about glia, and that gave us an excuse to contact R. Douglas Fields, who's a researcher at the NIH who wrote this book, and so we got to talk to him for an hour. I had David on the podcast, got to talk to him for an hour, and it's a great excuse to talk about stuff that you think is interesting, and then every once in a while you can use it as an excuse to rope an expert in and have an excuse to have an extra really cool person to talk to for an hour, and then you get to share it with everyone. And so part of what I like about outreach is that selfishly it's a lot of fun, and I love that I get to share my excitement about science. But Twitter, being on Twitter is totally a selfish experience for me because I feel like I benefit from Twitter in a big way. I've managed to connect with tons of scientists that I would not have known otherwise. Like Tommy, I think we probably met on Twitter.
Yeah, pretty much.
And I've met a number of other scientists that maybe I wouldn't have seen at conferences because they're on a different continent, and so the likelihood that we end up at the same conference is much lower. And then additionally, my killifish one year had a pathogen that I couldn't identify, and I put a photo of the pathogen up on Twitter, and within 30 minutes I knew what it was. And people told me what it was, and so I was able to treat it. It was too late for my experiment that season, but at least I knew what it was, and that saved me probably a month of time trying different treatments. I knew exactly what it was and how to treat it. So anyway, I find Twitter is a fun way to share stuff, but also selfishly, it's my favorite networking tool and my favorite way of learning stuff that would be hard for me to learn otherwise. So did I answer all of the questions?
Yeah.
There are a lot of questions about social media. All right.
Oh, before you go, Kelly, so if we want to sort of have a look at your blog or your podcasts or, where do we go?
So my blog is at weinersmith.com, WEINERSMITH, and I agree that's hilarious. And then my podcast is weeklyweinersmith.com, all one word. But the podcast isn't updating until my experiments are done in a month and a half. But there's plenty of old episodes to listen to if you're interested.
Fabulous, thank you. Tommy,
thank you.
Round up with the
I guess, do you want me to talk about my social media experience?
Yeah. So you bring it all together. Tell us how because you're at the other end of the of the hemisphere to a lot of people that you interact with. It must be kind of interesting and the dynamics are different because it's a very individual world, and you've got connections that you might not have had previously.
Yeah, well, Kelly and David are both people that I only kind of found out about through Twitter, and indeed I managed to connect with a lot of scientists through Twitter. The funny thing is that I didn't actually start using Twitter up until about 18 months ago, and up until that point, I was using Google Plus as a social media platform, but it wasn't until Google Plus that I started really getting into social media because I was blogging then. I was blogging starting from about 2010 when Susan Perkins started a blog called Parasite of the Day and was asking for contributors. And I contributed a lot because I really, really like parasites. It was like a kind of cathartic release, especially seeing how my friends can only listen to me talk about parasites so much until they kind of go a little bit crazy and go, Tommy, just stop with the parasites. And so I needed some kind of outlet to share to anyone who would listen about parasites. So that's why I decided to start writing a blog, and I started getting more and more, I guess, professional with the style that I blog. I started out with a blog post which is just a single paragraph down to the way that I write them now, which is always about a particular paper, and have them regularly out twice a month. And so through that, I kind of combined that with the Twitter aspects and the other social media aspects, and I just found that it was a very great way to both communicate science, but more of a kind of a networking tool that I guess people who don't use social media they kind of don't really understand that Twitter isn't really about broadcasting necessarily what it is that you're up to. It's very much for me as a networking tool.
Fantastic. That's great. It's so good to talk to you all, and thank you for participating in our first Inspiring Australia ASP Public Outreach Hangout on air. And hopefully we can reconnect, later on down the track to talk, to find out about, certainly about Kelly, about how your experiments are going on, and I really want to know more about the ants as well, David. Seriously, I think you could do a movie, a horror movie about the ants. But yeah, I really think that it's really amazing to find the parasitologists across the world, so willing to talk about and share their research, and just the interest that you all have for other people who don't know anything at all about parasites. It's just tremendous. Thank you. Tommy,
thank you all, and I'm very happy to come back at some other stage if you need to. And thank you, Tommy and Lisa, for setting this up.
Oh, thanks for coming, guys. Thanks, Kelly. Thanks, David, for coming and doing this podcast with us.
Yeah, I reckon Tommy, we should see if we can do one at the conference. We've got our ASP 50 years old in 2014, so we've got a conference in June. I'm just thinking, I wonder if we can do a big hangout at the conference, and just get a few. We can do it, maybe broadcast it.
We'll see how it goes then, and we'll.
Our technical level of should be improved by then, right?
Yeah.
Nelly, thank you so much. And David, thank you very much.
Okay, thank you. Bye.
Yeah. Thank you very much.
Bye, Kelly.
Bye.
Bye.